Actually, it seems like in some parts of this feature, the slope is actually more than 90 degrees, little bits of overhang just below where that Iridogorgia is. Amazing. Thank you.
No problem.
This just shows you how diverse the U.S. Caribbean is, just because yesterday we were on a really gentle slope, mostly composed of soft sediment, and 26 miles. We're in
very
this
nice
really
complementary light between the ROV and Sirius.
really cool topographical feature. This really steep ridge.
Definitely. Yeah, we are diving in a depth range we haven't explored before, and on a feature that was identified for multiple reasons, identified by our scientific community for multiple reasons for diving here. Partly because the Caribbean Ocean Time Series, or Caribbean Time Series, is in the vicinity, as well as a potential exploration target, because this area has not been explored for deep sea habitats that corals and sponges might provide. We don't anticipate on seeing a lot of commercial fishes as we've been seeing over the past few days. 2 days ago, notably, we saw several queen snapper. We wouldn't be seeing those at this depth. We're substantially deeper than a lot of fish species can be found. Off in the upper left corner, there seems to be a lot of branching stuff up there.
Now. Copy that. It's about 16, 17.
Yep. 4 to 5 minutes.
Over. Are we 10 meters in?
Yep.
Yeah.
Copy.
There's a lot of this mucus drapes on these rocks.
I've got my camera at 30 degrees, so.
Copy that. You can, yeah.
You heard
Turkey just centered up at this angle.
You heard the coral request. They wanted bottom left.
We have not made any collections today, mostly because we just came on this wall quite a bit just a few minutes ago, and you know, if there's anything that shoreside scientists find particularly striking, we can see what looks like collecting, but we'll have to.
Well, actually, did you ask to see something?
Yeah, there's something branching right in the very low left corner.
Copy that. There's a fish way back there.
Yeah, snap on it.
One of yeah.
Right down here?
Yeah, it's now camouflaged with the background, but it's right in the center.
It's right center of your laser.
Do a come partial video.
Branching. It's really small.
Oh yeah, well. It's tough to see this stuff. It's, you know, when you're looking straight onto the wall, this sediment drape is really making a lot of things blend into the background. Sometimes if you look at it at an angle, it's a little bit easier to see the motion of sea fans waving and stuff like that.
It looks like a bamboo.
Yep. This one looks to be a little bit more highly branched, or maybe just a larger colony than what we've been seeing. But I can see several anemones as well.
They indicate this is a
and looks like one of those egg cases also on this colony. That's what I'm seeing on the right-hand side. That could be a closed anemone.
Exactly.
Maybe we'll go ahead and call another 10 meters in.
Not sure what that is.
In 5 minutes we'll be done looking at it, and we can move up slope a little bit.
It could be an egg case.
Sounds good.
Same heading.
But yeah, a lot of small anemones on this colony.
Let's look at the egg case to the right.
Yeah, let's see here.
Just out of the screen to the bottom, I thought I saw some other associated organisms.
think a zero.
We could go down the colony.
The bottom. Copy.
070. We'll
Might
keep
be
it.
nothing.
070.
Okay.
Just more anemones.
10 meters, 070.
Yeah. That one branch tip seems to be covered with hydroids, but that's about it.
So when you're done here, Sean, let's go ahead and move up slope.
Yep. Watch leads. Anything else from
Full
this
stream.
That's it.
spot?
Would you, can we stay here? There's a stony structure above this colony.
Do you want to pause that move? Yeah, it's above this.
Yeah,
Yeah.
right above that colony there. Yeah, can we zoom in on that cluster?
If we're just zooming on it real quick, we should be okay.
Copy.
Got
Video
5 minutes.
snap.
Yeah, and those 10 meters will put Sirius slightly behind D2 anyway.
Tilt down and right towards the base.
So we're looking at now a bit more of a smaller cluster of this stony structure or a skeletal structure that's covered in manganese as well as some sponges.
You
I
want
don't think
anything
we've
else
gotten
from
a
this?
firm ID
Ashley?
on any particular sponges today. It's been pretty agnomatic.
You happy with that, Watch it?
Yeah, I think we can move on.
All right.
Let's
Thank
go
you.
ahead and move up slope.
Our depth now, we've made, let's see, we started at our dive at 2786 meters. Our depth now is around 2710 meters. So we're making a fair bit of vertical distance just in the last few moments.
Watch lead this now.
Test was what?
So watch lead, just wanted to
I
let
don't
you
see
know
it.
that right now the pace we need to make is about 0.1 knots, which is pretty quickly. Given the depth and the steepness of this feature, we can't really call long
I see.
moves. So we'll try to move as quickly as
Interesting.
we
Okay.
can. But also wanted to let you know that since we're so deep and the terrain is pretty steep, whenever you think we might sample, just let us know as
It's like
soon
odd.
as you hear
It's like
something
a-
from shore, because it will
I
take
keep
a little
adjusting my
bit to
Z
stop
bias
the
like
ship.
crazy.
Really? You think the
Yeah.
currents are kind of
Thanks for that
shifting
update,
as we
Matt.
move
We
up?
will be sure to
I,
let
like,
you know as
can't
soon as
settle.
possible.
Copy, thank you.
For the life of me. Auto depth was engaged. That would explain it. Nice video. That makes me feel a little bit better, but also worse.
Pilot, can we get a snap zoom on this anemone?
Yeah, copy that. I was getting close just in case you wanted it. Alright, video, let's come in.
Laser is clear.
Co-pilot, you want to grab? Thank you very much.
Not sure.
Sure what this, yeah, you could see this, I think the second ring of tentacles. Yeah.
So I missed part of your conversation, Nav.
Yeah, this
with
could be a
Watch Lead.
Syrianthid.
What was their determination for the pace?
No, I just let them know what
For
pace
some reason, this
we needed to
looks
achieve.
oddly familiar to
Okay.
me, so
Oh.
we might have seen it
I mean, I
previously,
told them
but
that we
it
can only
seems to be
make short
attached
moves
to the rock.
given the
Yeah.
so
terrain
I don't
and
think
the
it would be a
depth.
Syrianthid.
Looking
in
over
that
We'll try
case.
to get there,
They
but
don't.
Foothold.
we
They're
might
not
not get there.
particularly fond of rocky hard bottom.
And also to let us
But
know as
I can't
quickly
tell.
as possible whenever they think we
I can
might
see
sample.
that inner ring of
great.
tentacles, which is a real giveaway. And some
Let's
of
go
them
full
do
screen.
have tubes
Copy
that
that.
lay along rocks, or it may look like it's going into the rock, but there's some crevice or something that the tube is built into.
Right. Excellent. So we're looking at a dark Syrianthid
Okay, and that
anemone.
move was called in 1619. Is that right? Or 1620?
Yeah, I wrote down 620.
Okay.
All right,
Video's
pilot.
clear, so probably
I think the movie's done
satisfied with
already,
that one.
but,
Video is.
Video
Well,
whenever
Sirius.
you're happy.
Yep.
Sirius is
Great.
Copy. Thank you, Bridge.
Sirius is probably just at the beginning of that move.
Yep.
We are seeing a fair bit of sediment here, but much less than I expected. I was kind of expecting to see more of these drifts and sediment sand
can bring
channels,
lasers
or
back on.
sediment channels, but it's actually very rocky
Get it
and
for you.
not entirely smooth, which is kind of what corals and sponges would like. All of these boulders and small topographic relief in the wall is great at accelerating currents and possibly
Losing the skids.
resulting in enhanced food supply
Yeah,
for
feel free.
suspension
If you want
feeders like
video.
corals,
Is
Some past skids.
filter feeders
out
like
of depth.
sponges as well.
Stable enough to do shots, or you can
Not
do them?
usually. It kind of oscillates around a.
Looks odd, like it could
Yeah.
be trash or something.
It's
So
too
it's
white.
very
Shoot
steep.
that white thing straight ahead. I'll just zoom it from here.
Dead center. Can we zoom to the
Yeah,
other
go
side
for
of
it,
this
video.
canyon?
It's at auto depth. You'd probably be better
I'd
off
say
trying
that's
to zero
probably
your
trash.
Z bias.
Plastic bag, maybe?
Yeah, maybe. It's hard to see.
Most likely man-made. It's in a channel with a bunch of phytodetritus. It's sargassum. Okay, thanks, pilot.
No problem. Have video.
Coral whips on the upper right.
Copy that.
Yeah.
Said there might be it.
So on the top of this little pinnacle here, there seems to be a coral fan.
Copy
And
that.
if this is the same one, we may want to stop the ship to sample this
Ship stopped.
if
and
you're still moving.
Sirius is probably
Yeah,
pretty
this
much
might
settled
be
out.
a good time for that.
Okay. We would want to get a zoom
can come
first just to
down a little bit.
confirm, but we're
Yeah.
pretty sure that this is a potential sample candidate.
Copy that. All right, video. You want to start to come in?
Scott, you could let us know. I know this is a little bit of a smaller colony, but if you think this is the same one we've been seeing. There's also a black coral there in the background.
Yeah, one of the things, when we first discovered this, it was on New England Seamounts, on Nashville Seamount,
came
about
down in altitude a little bit to
2,200
this
meters
tether.
depth.
Copy that. Are you
And
happy
what
to
was
see
so
this?
amazing,
Good
this was
sample?
back
Yeah,
in, I
We're set
think,
up fine.
Great.
2004
Set
or
up nicely,
5, but the colony that we found had no nodes whatsoever. And so we were really scratching our heads trying to figure out what family it belonged to, if it was a Chrysogorgid or Primnoid or something else we didn't know. And then upon analysis of the polyps and the sclerites and the genetics, we found that, oh, you know, that's actually a bamboo coral. And we've since found others that do have nodes, and so we're not sure why those original colonies we found didn't, but really interesting. This one actually looks a little bit thicker than the ones we were seeing earlier.
It's got laser striped across it.
Well, it's not Okeanos, it's not branching at the nodes.
Okay, video's clear.
Great.
So I can't say with 100% certainty whether this is the same thing we were looking at below, but it certainly could be.
Yeah, it's definitely, it's reminiscent of that, the polyps, but the other one we were looking at a little bit lower seemed to have been a little bit more highly branched. Maybe it could have been older or just more well suited for that particular spot, but I think it's as good a chance as any to grab a sample here. Seems like okay topography for the pilots to land. And
so this is what you want, correct, Watchit?
Yeah, we're going to go for a bit of this.
Yeah, copy that.
And Scott, is there any interest in grabbing either a branch point or any particular sampling?
Okay. Yeah, 100%, because part of the taxonomy, although it's flawed, is based on those branch points. Steve, I know you know this. We have a lot of data that shows that, in fact, that's not a good way to separate out some of the higher level taxa. So, for example, at the genus level, separating Keratoisis from Lepidisis, you can't just rely on that branching. However, branching from the node versus the internode does seem to have some importance, and so preserving that in the sample is really useful.
Yeah.
So if that's possible, let's please do it.
Yep.
This is going to be really easy to snap or cut. It's relatively fragile, but it should, hold together pretty easily wherever it's sampled, and it should drop into the box fairly easily.
Stand by to just try to stick on this.
So, pilot, for the collection, we were going to relay some instructions. We're looking for enough of the material to get at least one or two branch points.
Can you help me keep an eye on the
analysis?
Sirius camera on your monitor there?
I anticipate this being very brittle and probably more likely a floater than a sinker.
Or the right monitor?
Copy. Brittle, and you want a branch point? Is that correct?
Yeah, a branch point or two.
Branch point or two. Copy.
I'm going to take both your cameras, Sean.
Yeah, copy. All right, Vida, you want to come in? Oh,
sorry.
You have my
wrong one.
Yeah.
Yep. There you go.
Thanks.
Bringing mini Zeus down.
All right, Vida, you want to come in partial? Let's try to find what part we want.
HD1.
Oops, sorry, I was then moving your camera.
Yeah, here you go. I'll just give it back to you. There you go.
So watch it. Maybe like left side.
Yeah, I was just looking at that. Left side looks like it could be better.
Copy.
If you grab low enough.
Yeah. Right underneath that branch point.
Yeah.
Does that work?
Yeah, I think so.
All right, great. All right, Vida, you want to come out partial? Yeah, I'll let you know when to stop. That's good there.
One thing that looks interesting to me is that this appears to have very long tentacles relative to
Copilot,
the
am I ready for
diameter
hydraulics?
of the actual
Yep, hydraulics are up.
body.
Which box are you going to try to stow it in?
Yeah, they do look very large.
Port outer. I'll see how much I get.
I was just thinking, based on the way that it's swaying in the current, could be an idea of how rigid the skeleton is, and it actually looks like the polyps have some sort of significant weight or drag on the branches, based on how they're moving. So the lasers are on the bottom for scale, folks on the shore, so you can see that this colony is not huge. It's maybe 30 or 40 centimeters, 40 centimeters, I would say, in height. and very, very thin. At best, I would say the base is half
you
a
want to center
millimeter or
up
so,
mini Zeus for me?
maybe a millimeter tops diameter.
And then video, can you get a partial
Or
zoom on
sorry,
mini?
centimeter, not millimeter.
Yeah, it's good video. Thank you.
D09. I think for the field, I
Alright. I'll move this over. Ooh, that one's pretty long, huh? It's alright.
So you want the port box?
Yeah, and then I'll probably have to...
Come up off the bottom.
Yeah.
Okay.
Both cameras full wide.
I'm going to bring your mini Zeus over to the box.
Thanks, video.
You might be able to just perch up on top there.
Yeah, nice. Just sideways a little bit. Little hop. Alright, you think that's good for drawer?
Yeah, I think so.
Alright. Go ahead and get it.
Okay, opening drawer. Okay, clear.
Yeah.
Retracting drawer. I'll follow you over with Mini Zeus to stow.
So that's a good collection. Yeah, Scott, you can breathe. We have a bamboo coral in our possession as the first sample for today's dive. Excellent collection work, pilots.
All right. Arm is off. All right.
Okay, Mini Zeus centered up. Camera back to you. Putting Sirius back in
So
monitor right.
that depth is about 2,700 meters.
Nice job, Sean.
Thank you.
Should we go ahead and get another 10 meters and
Our first collection of the dive.
Yeah. Gives us 5 minutes.
Yeah.
I think it's good.
Okay, 10 meters, 070.
All right, well, actually, was there anything else on that rock that you wanted to look at before I start moving away?
There's that black coral. But did you identify that black coral before?
We didn't get a chance to look at the black coral, no, but
We should
Great.
maybe
I'm
make
still.
way.
I could peek real quick if you want.
yeah, just a quick snap and then
Ready for
we'll
the
move
next
on.
move's gonna be 10 meters, bearing 070.
Yeah, just
speed
behind
0.2.
the coral we sampled, I did see a black coral colony, and it was
Good copy.
had very clear translucent polyps.
All right, Vida, you want to come on in, snap it?
Did it move?
I don't know.
It was here, wasn't it?
Yeah,
Yeah.
I remember
Well,
seeing it.
Maybe it was in the back.
Is that the
all
same
right. Well,
coral?
yeah. Mysterious ghost black coral.
Just go center and I'll go all the way in. Center it up a little bit.
Yeah.
Yep, I don't see it.
Weird because I saw it when we were collecting
Me
it.
too. Yeah.
I don't think I grabbed it. I think it was further away than.
Well, let's hope you grabbed it.
I did
Well,
not
a
see
lot
it.
of happy scientists, I'm sure. I don't think so, but that's a great collection anyway.
All right, thank you. All right, moving on.
I think that'll be the first coral associate of a coral we ever sample. If they are attached. It's not likely.
Yeah, it's really hard to see. You have to look at side view to see a lot of these branching corals because they
Yeah.
really blend in with the background.
Yeah. That's why it helps to look along the wall once in a while, because you see things with this the three-dimensional view that you don't quite see when you're looking at it straight on. I think the most fascinating thing for me is these massive stumps. And it's always a big mystery when you see biology, stuff that's obviously biology that you're not seeing the live version of. Because there's a lot of debris in this area.
Yeah.
Depending on how far we get, I think this debris could be interesting to look at too. And if it still is there when we get a little bit higher up the slope.
a little bit of an overhang, it looks like.
Yeah, copy that. I see it.
Buenas tardes a todos están escuchando. Hoy estamos explorando
I think I'm starting to move now.
Copy
una
that.
cuesta
About 4 minutes.
al sur de suroeste
Copy, Bridge.
de
Thank
la
you.
costa
Last one
de
complete.
Puerto Rico. Se llama Jaguey Spur. Es más o menos 24, 26 millas afuera de la costa. Colectamos un coral blando, se llama coral bambú. Y vimos muchos corales negros y blandos en este buceo. Y también esponjas de vidrio.
Looks like it's
Ahora
starting to
la
flatten out a bit,
profundidad
Sean. We're starting to see pretty
es
far ahead of us here.
Yeah.
Couple of interesting features
2.698
here, maybe.
metros.
It's harder turns.
Empezamos a 2.700 metros de
Maybe we should
profundidad.
put in another move. What do you think?
I think we could move 10 meters, another, no problem.
Looks like the slope is moderating a little bit up here.
Um,
We just went up a rather steep
how
wall
far is it? That's
with
like
at least 3 or 4
meters,
colonies.
so 10.
4 colonies by my count of
It's 10 might
Iridogorgia
put you where
magnispiralis,
D2 is, right?
Right.
which is a very large
And-
spiral coral.
Yeah, if you want 10 meters,
Or
fine.
helical.
let's do that. Let's do another 10
coil.
meters, Fernando.
Same bearing, 70.
Looks like something rolled down here.
Yeah, 070 looks good.
Copy.
It looks like a debris chute.
Video, you want to snap right here? Is that anything?
Bridge, does it have?
Nope.
Yeah, it just
some of
looks
that.
like debris of some sort. A sargassum.
Sargassum and coral debris.
Oh, you can come out video. Thanks.
I think this type of habitat is perfect for corals. I think they really love these boulders that jut out from a steep wall. And you can see the current, at least the particulate matter in the water column is moving fairly quickly. But that could just be our movement too. But I get the feeling that since we've seen such large colonies, the flow conditions are good enough for some of them. If you're just tuning in, we're diving on the southwest part of Puerto Rico, offshore about 20 miles or so, in an area called the Jaguey Spur, and this particular deep sea feature is an extension
Yeah, do what you're doing. Maybe explore this rock for a minute. I'll let you know
that
when
juts
I'm feeling
out
that last
into
move.
the Caribbean
Sounds
basin,
good.
and it's suspected to have rather abundant hard bottom. We mapped over 2 nights ago to look at the seafloor bathymetry in this area, as well as some characteristics about the substrate. And we found that there was a mix of hard bottom and soft bottom here. So it seems like we've hit the jackpot in terms of hard bottom. This is really good stuff for corals and sponges to attach to. Not much in the way of fishes, though.
That's nice. That sponge hanging out.
We snap zoom on that stalk, that white thing up at on
yeah,
that stalk.
copy that. My video is at centers. You want to snap?
Yep. Co-pilot, can we leads ROV just a little bit on that shot?
Looks like a little baby
Pretty dramatic from your
stock
view.
sponge.
Stalk sponge.
Don't want to turn too much because we're moving in this direction right now. I'll
Ah,
give you a little
they're
view.
tiny. Look like tulips.
Yeah.
Glass tulips.
Copy, Bridge. Most
Yeah,
complete.
everything seems to be covered. Oh, I see some of these attached, or some of these stoloniferous octocorals that we were seeing a little bit deeper too. Did you see those when we first
No,
No.
I must have
It's
been
a-
at lunch.
Yeah, small type. It's not what's making the skeletons, but it's a small stoloniferous octocoral that we've seen on some of this hard substrate. They don't get much bigger than that, unfortunately.
Right there with the polyps out?
Yep.
Yeah.
Video's clear.
Copy that. Watching anything else from this spot?
That's that.
All right, sounds good. All right, video, you can come wide.
Are those the bases that we're seeing?
I don't
Yeah.
think so. No, those bases belong to something large. You know, there are bases like that that could belong to corals, like bamboo corals can grow that big and leave these substantial bases, or paragorgiids can grow that big, as well as occasionally some primnoids can grow fairly thick, but my guess is that it's probably either a bamboo, or historically could have been a bamboo coral, or maybe some kind of, could be a corallid too. It could also be a sponge, you never know, but these look pretty hard.
If I were to
Steve,
rub
can
my
we get a zoom on this anemone-like
Yep.
thing? I just want to rule out that it's not relicanthus.
Pilot, can we zoom on the anemone?
Yeah, copy that.
if I
Yeah.
were to rub my hand against those outer tentacles, will I feel a sting?
Don't tempt us, Roland.
No, I just want to know, are those stinging cells on the outer one?
All right, video, you can see.
So
comment.
by definition, anemones belong to the phylum Cnidaria. So
Goodbye,
this particular
interlacers.
anemone is a cnidarian, and all cnidarians have these stinging cells. This one looks a little bit different.
Definitely moving.
You
little
can see
more
the end
leash.
of the ring, I believe. Yeah.
The stinging cells are.
Another stereanthus anemone, another tube anemone. Thanks for checking.
It looks like part of the body might be in the crevice of this rock, which is similar to what you were saying before. They might be lying along the rock rather than inside of the sediment.
I think we're clear.
Copy that.
Thanks, pilot. We can
No
continue.
I'm thinking if
So
settle out
the
here for a
difference
little
with
bit.
these,
maybe this is
with a
something
lot of
interesting
this
over here.
stinging organisms, is
Yeah,
that
copy that.
So after
Come
your
back
port.
center.
different cnidarians might have different size, harpoon-like
I can
structures that
turn my head to follow
are
over there.
Copy.
the stinging arm of the cell. But at the same time, you know, depending on what type of organism you are, if you're a gelatinous organism or if you're a fish, you're going to experience a different sensation than if you had a very thick epidermis or skin like you know, we would. So if you rubbed your hand across it, you might not necessarily feel anything.
There's the fish in Sirius's
Cool.
camera.
Yeah.
Or it could be extremely painful. That's something you'll probably
zoom on
remember for the rest of your
Sure.
life.
Hold that.
Fish and Sirius.
Steve, are you speaking from experience?
Okay, let's come out.
I haven't been
See
stung
himself.
that poorly, but I have to say that I have been stung by some deep sea siphonophores, and they really, really hurt.
I've been stung by
I've been stung by
cubozoans.
the irregular urchin spines, and that is not pleasant at all.
Yeah.
Oh, the fiery urchins? The fire urchins?
They weren't fire urchins. It was a deep sea urchin. I don't know that it had a name, and we collected it. And
The
I grabbed it and
formosoma.
got one of those spines in the knuckle and remembered that for the next week.
Oof.
Yeah, every time Chris Ma comes in to tell us about the pancake urchins, the formosoma urchins, those are extremely venomous, and they can leave a very potent sting if they were to touch you. No, but you know, we handle octocorals and things we collect on the seafloor with gloves usually, so we don't get stung when we handle these on the deck.
I think I'm looking like right at it right now.
Well, I know the black corals on mesophotic reefs, they really hurt when you touch them. They definitely leave a mark on your hand.
That's interesting.
Would you think this coral rubble we're seeing here is from some catastrophic event or just some long eon type of movement?
No, it's hard to tell what the rubble is here. You know,
Do you want another 10?
whatever happened, it happened a long time
Yeah, what do
ago
you think,
because
Sean?
there's a significant amount of
Here for a few minutes.
iron
maybe 5
manganese
minutes before
crust
I even feel the move.
on a lot of these skeletons, but
Yeah.
Do 10 meters at
they also
070
seem to
again.
be recolonized
Copy.
Yeah, sounds good to me.
by sponges or bryozoans or other organisms like that.
Here's something branching.
Oh yeah,
Oh,
wow, cool.
whoa.
This is now.
There's, have you seen this before?
We saw Chrysogorgia a little bit below, but this is much bigger.
Video, you want to start to zoom?
Can we zoom, please? Yep.
It's really hard.
Might have to get really close to this. It's got
It's big.
extremely small polyps.
Copy. If you want to
So,
get closer, just come back out
This
before
one,
I move.
this one looks different from the last one, but likely only in the way that it's not covered with this kind of mucousy sediment sheath. You can definitely see the golden color axis on this coral that's reminiscent of the family name Chrysogorgia, Chrysogorgiidae.
Bridge. This is now.
This could be in the genus Chrysogorgia, but we'll get a
Alright, video,
better
start to come
look
in.
and see if we can find any characters that might help us make that identification. Although it's not a morphology I'm familiar with. This one is very much fan-shaped, very large, planar fan. Not too much three-dimensionality here.
Looks like a honeycomb. Has these weird
They have
branches.
a very distinct branching pattern. That's actually one of the characters that can be used to identify
Bridge. This is now.
corals in this genus, Chrysogorgia.
Ready for the next move. Same as before. 10 meters. Bearing 070. Speed 0.2.
Very nice.
How deep can Chrysogorgia go?
Good copy.
Oh, I'd have to look and see what the depth record for this genus is.
Yeah, I'm about settled out now, Sean. So
I think we were
5
observing
minutes
them in
before
the
we feel the move.
Pacific down
Ready for
to
full screen?
Yeah. Go for it, video.
2,500 meters or
Okay.
maybe deeper. But yeah, this is pretty deep.
Lasers off.
I would say they can be
Right
found
away.
deeper,
Yeah,
but
thanks.
we often don't go
Tilt up.
beyond 3,000 meters if we're looking for coral habitat. But
It's bigger than
I'm
I thought.
fairly certain they can be found down there. But it's not something you would necessarily see in abyssal environments or even
Got it full
ultra-abyssal
now.
environments, but it's good to document
Say it again,
this.
video.
I think we've got it full now.
Oh, copy that. Yeah.
Thank you, pilot.
Pretty wide from top to bottom.
Yeah. All right.
Full wide.
Copy. Moving on.
I'll have to look at the statistics on those corals, their depth distribution. I don't think it's
Another
unreasonable to see them
3.5
at this
minutes
depth,
before we move.
but I'm just not sure how much
Copy.
deeper they extend to.
Yeah.
A lot of the data we have for many of these coral families is, extends to maybe a couple thousand meters, but beyond that, there hasn't been a lot of work done. So we really don't know what the deeper end of a lot of the depth distribution for these coral species is.
Oh, it's like this pinnacle that we're.
There's a lot of debris here. So
Yeah.
you were asking about this debris, and it's really tough to say. I think it's probably a matter of time, seeing that it seems to be more or less set in place here and fairly well coated. looks like a sponge, right?
Yeah,
Yeah.
it looks like one of those small stock sponge.
Sometimes those sponges or light colored structures can sometimes resemble sea stars. It's always good to get another angle on it, especially if the sea star is feeding. Sometimes their symmetry is a little bit
Yeah, they're
modified.
more like bulbous
Yeah.
looking.
This area looked like it used to be extremely coral popular back in the day.
Yeah, that's what I've been saying for a little bit, that a lot of these
Copy bridge.
branches,
Complete.
they're probably quite old, and iron manganese coating like this accretes at a rate of a very slow rate, millimeters per 100 years, I would guess, or 1,000 years. So quite old.
Where do you think the sediment is from? Because we're far, I mean, where it has originated.
Yeah. So I was thinking a couple things. It could be, so we mentioned that this area is a target area for South American river plumes.
Yeah.
And, maybe there's some sort of sedimentation deposition that occurs in this area from those plumes.
So you think you're moving?
If the signal reaches this point. But more than likely, it's from oceanic. It's sediments from oceanic processes like deposition of shell material and phytodetritus and degradation of that material on the seafloor or in the form of marine snow.
Ooh.
It's very powdery. Yeah. I think we might have just found our answer.
Whoa, that's impressive.
I think we should get another 10 meters in.
I was worried that this was just going to be like a graveyard of old corals, and we're like, sorry, you're a couple thousand years too late. You'll never find out what's
we're
here.
like halfway through that last
But
one.
I
Get
think
another
we
10
might
meters at
actually
080.
be
There's a
seeing
couple,
here what this
yeah.
is.
The lasers on.
Looks like there's two
Yeah.
different.
Then we can
Or
get over to
whatever.
that coral.
Yeah.
Okay, let's do it, Fernando.
Right.
This might be one of the largest coral
Great
fans we've ever
design.
seen on this expedition.
So we'll put
Next
lasers at
move is
base.
gonna
that'd
be
closer.
range 10 meters, bearing 080 degrees, speed 0 decimal 2.
So I don't know if you have. Well, you got some room to push forward. I
Pilot,
don't know if you
could
can
we
quite get there, but
stay here for a couple seconds just to look
Good
at
copy.
these? I don't know how the ship is moving or.
We just put a move in to
Okay.
get closer to these. So
Oh,
yeah.
perfect. Ideal.
I can get closer, but just give me one sec.
yeah, you might be able to get out
This
there, and
is
then I'll be moving to
Very
in a few minutes.
fascinating stuff.
Yeah.
This is the point of the dive where, if I'm watching from the shore, I start throwing all sorts of photos up on Twitter and
Might wanna
social
take a
media.
quick snap to make sure we're on the front side of it.
Sure.
Right now we're
Up
at
on
a
the
depth
front
of
side.
2,680 meters.
Yeah, you've definitely got tether, Sean.
Amazing.
And we'll just get a little closer to you. Make it easier.
So this to me looks
We
like
got enough
an
detail
entire
on this side.
Great.
family of corals we haven't seen yet on this expedition. I'm guessing both of them, the one in the foreground and the one in the background as well. This looks like,
So we'll start with
just from a brief glimpse, it
Yes,
could be a
right on the big
coralliid
one.
or a precious
Start on the
coral.
lower section with
That's
the
start lower section.
extra fuzzies.
Copy.
And these precious corals, we were seeing them
Going on that.
extensively in the Pacific, but they're just not very common here in the Atlantic. And
Lasers are clear.
All right. Go
we
back.
don't know much
Grab
about them in
lasers
this
and
part of the world.
Thank you, sir.
Slip left of lasers.
There's a lot of attached
There's
stuff
no
here.
lasers anymore.
Yeah.
You could spend all day going through this.
Oh, there's some egg cases. It looks
Barnacles.
barnacles.
Yep, barnacles. That looks like a closed anemone right there on the screen right now. As we scan through this colony,
Scan diagonal upright. Slowly.
I
Diagonal
might go as far
up,
as to say as this
right?
looks
Yep.
like corallium.
Copy.
There are only only a few species of corallium that are known from this part of the western Atlantic. Probably, let's see.
Oh, look at this is interesting. Like it
Has some damage.
Yeah.
Maybe
Covered
it's
bridge,
regrown.
not complete.
Yep.
here's also like a node type branching. It's weird.
Yeah, we see this sometimes on octocorals,
Pull you
where
out a little bit.
if they have some sort of injury, they can either grow
So I
over
send you a video?
Going to pull you out a little bit.
whatever
Oh,
is
copy.
causing the injury, or grow around
Some
it,
pink
and
things up or
end
left.
up with these bizarre morphologies in their
going
skeleton.
Down center. Looks like a nice start.
So it looks like we're looking at a correlated fan here, corallium possibly. It's definitely the depth range for these coralliids in the Atlantic.
Es un coral blando en la profundidad de 2.680 metros.
This is pretty deep for coralliids. I would think to see them a little bit shallower, but the deepest record I have from the Anegada Passage is around 1,500
Definitely moving
meters.
now.
Oh wow,
So
pulling out.
1,000 meters deeper.
quite deep.
Oh wow, that wasn't even
There's
to
some
the base
really
yet.
big anemones,
So
so
for
this
sure it's getting to the top.
looks like it could be
Yeah.
pretty old colony.
Yeah.
By the size, yeah,
Oh,
definitely.
absolutely.
Go down to the base.
Base, got it.
Fascinating.
Yeah, it's very
Drifting
beautiful.
back.
There's I thought I saw a couple of other things in the background as well. After we're done imaging this,
Yeah, copy that.
maybe getting lasers on the base if it's possible.
Wow, look at that! How big the base is.
Yeah, we'll get lasers after we zoom out from this.
Can you rotate right?
See a bunch of these amphipod tubes at the base as well. These organisms look like they're making these mucus tubes or these sediment tubes. We've been seeing them over the past 100 meters or so. We've been climbing up. But that is one big base.
Yeah.
Lasers
Kind of
on.
the size of the base we were
Base
seeing on some of these
flying
large
Sure. Lasers
colonies
on.
in the Pacific. Yeah, you can see these kind of galls
10
that are
centimeters.
occurring.
So yeah, lasers are on right now, watch
Wow.
these.
Yeah,
It's one big base.
at least 10 centimeters across, probably
Yeah.
more, just based on our viewing angle here.
Look at those white bulbulous.
Pretty
just
awesome.
Diagonal up left from lasers.
All right,
Wild looking feature.
Pilot, I think we've made a good identification on this colony.
Copy that. So you're happy with everything then?
We're happy
Good
here.
move
In the
on.
background, we thought we saw something, if there's any more tether to get over there. I know it's
Yeah,
a
just
big
be
fan to
mindful of
try
that
and navigate around.
Point behind you to your port.
Full wide.
Oh, to
The
yeah.
one we just came up. It's not too far behind
Yeah.
D2's tail.
Oh, like in the background, watch it.
Yeah, there's a large sponge and a large coral in the background as well.
Definitely get over there.
Yeah.
Morphology.
That.
If you can get it from a distance, that might be acceptable too.
Is it the one dead center? Is that what you
Yeah,
want?
the two. Yep.
All right, Vidya, you want to snap this?
Ooh, it's a different sponge.
Yeah. So this kind of resembles this horn-shaped sponge we were seeing a few days ago on the trough.
Yeah.
You remember that one?
Yeah, that was pretty common.
It was on the vertical face, also. Seems to be there a long time. There's a lot of dead sponge material.
Is that all you need from that watch?
That's great. We can move on.
Copy that. All right, Vidya, whenever you're happy.
Looks like quite a face to your starboard, and it looks like there's another fan.
Oh yeah.
We want to work our way up there. Or maybe it's a big sponge. Can't tell.
Okay.
And I'm pretty much settled out now, so.
Yeah, copy that.
Nicely done, pilots. I think you guys solved the mystery of what those giant footholds were down below.
This looks like
Oh, that's a cool feature. Look at that crack.
Yeah.
Might be. It looks like a sponge stalk.
Yeah, maybe with zoanthids.
So
Do you
anthids
want to
are
zoom,
some of
Swashits?
their fouling. Maybe snap zoom at 6 o'clock. Yeah. Nothing we need to dwell on, but just to get a good confirmation. Oh, is that the yellow
Yeah. Oh no.
octocoral video? I think this is the one that yellow octocoral here that we saw just behind the sponge. The video mentioned that there was a yellow.
It wasn't quite as many as there are here.
Yeah. So this looks like it could be a stoloniferan as well. We were seeing something very much like this, but they were very, very much clear and not yellow like this, but it could be some sort of color dimorphism. So good to document that. Yellow and clear. Could be a different species as well, but given the similar overall colony morphology, I would say it's probably just different colors here. It's very frequent though. This one's covered with them.
Yeah. The bamboo coral also.
Yep. Now that bamboo looks similar to the one we saw a while ago. Might be the same one we collected.
So there's spiral coral on the right.
See in Seriosphere there's some stuff coming up too. Another Iridogorgia.
I'm just going to peek up with my camera real quick.
You got it.
I mean,
Cool.
with the exception of that last corallium, Iridogorgia was the largest, but I think that colony may have been bigger. Maybe not longer necessarily, but definitely in terms of dimensions, wider and taller. Although, you know, I still have a shred of doubt that some of those large bases we were seeing that have this black crust might be all corallium. They could be something else too, so it's always a possibility. Could be something else.